Good morning, good afternoon, and good evening, everyone. And welcome to this Crown Bioscience Scientist.com and Inside Scientific webinar titled Targeting T Cells Within the Cancer Immunity Cycle. This is the second of 4 webinars in our series covering the methods, challenges, and opportunities along each step of the cancer immunity cycle. I'm Liam Sanyo for the events team here at Scientist.com, and I'll be the host today. From Crown Bioscience, we're being joined by Dr. Marrit Putker, who's Director of In Vitro and R&D. Dr. Saskia De Man, who's a scientist, and Dr. Pirouz Daftarian, Director of Scientific Engagement. Their presentations will focus on the impact of T-cell-directed immunotherapy on the cancer immunity cycle, mechanisms and targets capable of modulating and enhancing T-cell activity, and the application of relevant models and methods, including organoids, co-cultures, and the integration of high-content imaging. And with that, I'd like to welcome our first speaker, Dr. Marrit Putker. Marrit, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, thank you very much, Liam, for this very kind introduction, and welcome everyone to this second seminar in our Cancer Immunity Cycle seminar series. My name is Marrit Putker. I am indeed leading Crown Bio's in vitro R&D activities. I am based in Utrecht in the Netherlands, where we focus on developing advanced preclinical organoid models and assays, which is also what I will be covering today in today's seminar before heading off to Saskia. During the series and during the seminars in the series, we try also to learn who our audience is and try to adapt a bit towards the audience. And for this purpose, we ask the questions. And in the last seminar, we asked the question, on which level are you familiar with the organoid technology? And we actually learned that almost 50% of our audience was not yet familiar with organoids at all. And also a large proportion of people were actually interested in learning more or maybe interested in moving into using organoids, but did not have any experience with it themselves yet. So therefore, we decided to make use of the opportunity to give some more background on the organoids in this seminar, before then going into how they are actually interesting models in modeling the T-cell responses. So when we talk about organoids in the context of preclinical drug discovery, we can in fact view these models as in vitro patient avatars, which allow a very accurate modeling of tumor drug responses. So they show a very high degree of similarity to the tumor that they were derived from, which I will show you in a second how we do that. And this similarity extends both phenotypically and genotypically. And on top of that, they are very genetically stable, which makes them a very representative and very reliable models of the original tumors. And aligning with this, there is now more and more evidence emerging that these models have a very great predictive power when it comes to the drug responses when we compare this to the original tumor, which is what makes them also so interesting for preclinical drug development purposes. Now, at the basis of the organoid technology lie these adult stem cells, which are present in each epithelial tissue to assure a healthy renewal. And when we take a piece of epithelial tissue, either from normal or diseased tumor tissue, and we incubate these isolated cells in the right growth conditions, containing the right growth factor cocktails specifically designed for particular indications, and embed them in these extracellular matrix components, we can actually stimulate these stem cells to grow and to form these 3D structures, which we like to call organoids. Now, the fact that these organoids are derived directly from the tumor stem cells and are kept in an environment which is as native as possible also explains why these organoids have such a high degree of resemblance with the original tissue and also their drug responses, and also allows them to be grown almost indefinitely, making them very easily scalable, and therefore these models actually provide a superior efficiency and reproducibility when compared to many other preclinical models. Now, I already touched on this briefly in the previous slide, but I would like to stress here also the uniqueness of this technology, because it actually allows the development of matched normal and tumor organoids, as shown also, for example, here in a subset from our colorectal biobank. So these images were actually made in our high-content imaging platform that we also use for our drug screening purposes. And here I just wanted to illustrate all the different morphologies that these nice 3D structures can take. And here, all of the tumor and normal organoids are actually lined up on top of each other. And what you can hopefully appreciate is how all of these normal organoids look very nice and similar all with cystic morphologies and with a very clear single layer of epithelial cells with a one single lumen per organoid. In contrast to these tumor organoids, or we show here an enlargement as well, which show a very variable morphology, which often resembles the structure also of the original tumor that they were derived from. Now, here already I show you only a small part of our biobank, and you can already appreciate that we have a variety of different driver mutation in these different organoid models. And by banking more and more of these models, we aim to, in the end, have like a representation of the full spectrum of cancer in our biobank. So we have two sources of material to build our biobank at Crown Bio. We use these directly from the patient isolated tumors to derive patient-derived organoids. We source these in. We have these of at least 6 different indications now, and this is also still expanding. And these PDOs, they allow this generation of the paired models of, for example, the primary and the metastatic models, but also, as I just explained, the normal and the tumor matched pairs. And on top, it also allows the harvesting of other matching tissues, which I will touch upon later in this talk. The other source of organoids, we're using the PDX-derived tumors. We call these organoids PDXO, PDX-derived organoids. And we generate these from our large bank of tumors that we have in our PDX biobank. So here, of course, we do not have the opportunity to generate matching normal organoids, but we do have the matching in vivo models, which allows a different transition in the drug development pipeline. So, for example, it allows to select a large selection of PDXO models, to do a large screen and to narrow down the model selection there. Or it allows, for example, to do a large screen with many compounds or many combinations of compounds to then tune it down for the in vivo studies later onwards. So here's a snapshot of what our current biobank looks like, with over 500 different models spanning over 17 different indications. And we're adding novel models on a very regular basis to try and provide the most complete set of preclinical models covering all different subtypes of cancer with as many different genotypes as possible as well. So we do a very thorough and just characterization of all of these models. And I also strongly invite you to actually have a look at our website, make an account to our database, and to browse through to the database and see if there's any models of interest to you. Now, how can we then put this organoid platform to its best use? And here we show a very nice example of what this organoid platform can do actually. So it of course allows the differentiation of responses from different models in an in vitro setting. And for example, in this study, which was published this year, we used a couple of organoids initially to narrow down a selection of over 500 different compounds to select only a handful of candidates to move down in the pipeline, where we tested the subset of compounds on a panel of over 20 organoid models, all colorectal models, to study the differential responses of all of these organoids, and to in the end identify a lead compound which has a very high selective activity to the tumor organoids with a very low IC50, also compared to the control antibody, in this case the cetuximab antibody, and also being very specific towards the tumor organoids and not affecting the normal organoids in the dose range that we are aiming for. So next to this lead compound identification and being able to distinguish responses to tumor organoids versus normal organoids, we also were able to do some mechanism of action analysis by using this high content imaging analysis of the receptor responses, and next to that also do some biomarker analysis by distinguishing the responses, in this case, for example, on the percentage of the LGR5 cells in the model, which predicted whether a model was responsive to this MCA158 bispecific antibody or not. Now here in this case we used a FACS-stained biomarker, but this we can of course also do on using the NGS data that we have available for all of our models, illustrating the versatile possibilities of these organoids studies. Now the predictive power of these organoids we can of course also use when studying the cancer immunity cycle in, for example, these co-culture assays that we're showing here. So here we have actually co-cultured 5 different organoid models, either colorectal, ovarian, and lung models, with either naive in dark red or pre-activated PBMCs in light red. And we followed the responses of the organoids by looking at the size of their volume. And what you can appreciate is that indeed these different models have a very different sensitivity to T-cell mediated killing. If we focus here on these 2 left colorectal models, they were incubated with the same PBMC donor cells. And you can appreciate that whereas this model is very sensitive to immune cell mediated killing, this second model is actually hardly sensitive at all. Now these differences are very interesting to model. We can add more drugs, of course, to test if we can modulate the responses. And we can combine this as well with mechanism of action and biomarker analyses. This is part of our daily business, and Saskia will also go into more depth on these assays. But what I like to show you here as well is some of the work that we're currently developing, because where we are in the previous experiments actually looking at the allergenic response of these organoids to the immune cells. The organoid platform also allows us to start modeling the autologous response, because we can, while harvesting the patient material, the tumor, also harvest the matching blood, the PBMCs, or even try to establish TIL cultures, so the tumor infiltrating lymphocytes, then expand these TILs and try to perform co-cultures there. And well, as shown here, a nice overview of all of the advantages and disadvantages of the autologous versus allogeneic cells in a CAR T cell setting. These advantages and disadvantages also make a lot of sense actually for our platform. So where the autologous cells have a low immunogenicity in the antigen A-specific manner, the big advantage, of course, is that we can actually start to model also the antigen-specific immunogenicity, because these TILs are also expected to have the tumor antigen-specific targeting T cells. The downside is, of course, that there's limited material, so it's a bit lower throughput and it's a bit more expensive, but it's still a very interesting angle for our immunity oncology platform. So this is why we're trying to develop a biobank of patient-matched PBMCs and TILs and organoids. So this is the experimental setup here. So we derive the organoids and the TILs from one tumor. We bank also the PBMCs. We expand the TILs by a rapid expansion protocol. And then we, in different combinations, do the co-culture experiments and do a readout with a high content imaging in our high content imaging platform. And here is some work in progress. I'm just showing some early results. And when we look at these 2 models where we've also banked the PBMCs, but also the autologous TILs, we can in these cases actually see that there's not that much additive effect of using the autologous TIL fraction over the allogenic TIL fraction. So we see similar responses in this case. And what you can also appreciate, especially with this left model, is that this model is pretty resistant to immune cell mediated killing, which may actually be a very interesting finding because it allows us to test whether the killing was actually inhibited by the immune checkpoint. So this is what we set out to test immediately after. So we took these resistant organoids and performed a similar experiment, co-culture experiment, but then in the presence of different checkpoint inhibitors. Unfortunately, we were not able to identify a response here. Now, obviously, in the clinic, only 20% of the patients actually respond to this type of therapy. So that could be an easy explanation of not finding a response here either. However, next to this, it could also be that we do not have enough of the antigen-specific T cells in our TIL mix. So if you look at this TCR characterization of another similar TIL sample that we've recently banked, you can nicely see how polyclonal this TIL population actually is. So if then the antigen-specific TIL population is very small, it's most likely that we cannot kill the organoids with this small population. So therefore, we are now aiming on looking at enrichment of these reactive TILs first before adding them into the mix. And already some very interesting protocols have been described in literature. This is a very interesting one by the Dijkstra protocol, where they were in fact training the circulating T cells from a patient with the organoids to become tumor targeting T cells. And this indeed increased the activation of these T cells that were harvested from the blood, and also increased the killing of these T cells in an antigen specific manner. So we could block it by an antibody blockage of the MHC1 protein. We have actually slightly adapted this protocol to be able to enrich the tumor-specific TILs from our already established polyclonal TIL populations by co-culturing the TILs in the presence of our matching PDOs for about 3 weeks, and by then selecting the activated cells from this population by magnetic cell separation. So here you can see the TIL TCRs before this enrichment, and what you can appreciate that indeed after the enrichment we find a very more clonal population of TILs. And when we use this positive fraction in a co-culture assay, and here I'm only afraid I can only show you the bright field images so far, but what you can appreciate it if you look at the controls of the staurosporin where all of the organoids are dead. Here are the PDOs without any co-culture. Then the polyclonal TILs are not doing that much to these organoids. But then if we add the monoclonal enriched positive fraction of these TILs, we can see that these organoids resemble the staurosporin morphology, indicating that these organoids are indeed being killed by this fraction. Now, this is very preliminary data, so we're moving into our high content imaging platform to develop this further, and we hope to be able to show you more of this work in the near future. So with this, I would like to thank you for your attention and give the floor to Saskia, who will go deeper into all of the other ways of looking at T cell activities in the in vitro setting. Thank you.
Well, thank you so much, Marrit, for introducing the organoid platform and introducing me. So my name is Saskia De Man. I'm a scientist in the immune oncology department of Crown Bioscience, the Netherlands. And today I will dive further with you into testing of T-cell therapeutics in vitro with a focus really on high content imaging in our 3D platform. So for those of you who have seen the previous webinar, this figure might be familiar. This represents the cancer immunity cycle, and it starts all with the release of antigens through different processes of cell death, which can lead to the priming of antigen presenting cells such as dendritic cells, followed by T cell priming, and those T cells can then migrate, infiltrate, and recognize the tumor and lead to tumor killing. However, in cancer, many of these steps can be inhibited or evaded by the tumor and the tumor microenvironment. Luckily, we have, as a scientific community, already developed many therapeutics that reactivate this cycle. And I have highlighted 3 here, where I will talk a bit more about today. So these are T-cell engagers, engineered T-cells, and checkpoint inhibitors that help in re-stimulating the recognition and killing of the tumor. There are already several of these that are being used in the clinic and have shown great success. For example, Keytruda, or also known as pembrolizumab, and atezolizumab have seen great success as checkpoint inhibitors. Furthermore, cellular therapies such as Kymriah, which targets CD19 as a CAR T cell, is used regularly in refractory follicular lymphoma. And also other cellular therapies such as Provenge, which is actually a cellular cancer vaccine, can activate T cells to recognize a specific tumor antigen, and this is regularly used in metastatic prostate cancer. Also, for bispecific antibodies, there are already several, like Blincyto, which targets CD19, and Tecvali, which targets BCMA, used in the clinic to treat hematological cancers. And of course, it doesn't stop there, because we still have many ongoing clinical trials. So this figure shows that in 2020 there were over 4,000 immune modulators such as checkpoint inhibitors, over 900 trials for cell therapies, and also over 100 trials for CD3 targeted bispecific antibodies, showing the relevance of T-cell cancer. And now I will dive a little bit earlier in this discovery process. So how can we in vitro, in preclinical research, research these processes? I will first start with an overview of commonly used techniques to see the T-cell activation. These include, for example, FACS analysis on T cells of activation markers such as CD69, and also of proliferation, for example, here measured by CFSE, also with FACS analysis. Furthermore, we can also measure the cytokine release to see the pro- and anti-inflammatory cytokines and chemokines that are released. There are also assays, such as this one, that can directly measure TCR activation based on the engagement of the TCR itself, and also inhibitory and stimulatory factors, which leads to the expression, for example, of a luminescence molecule such as luciferase. Finally, a bit more towards killing already, we can also look with microscopy, for example, at the formation of immune synapses with microscopy, which is also a measure of engagement of the T cells and their efficacy. For many of us, however, cytotoxicity is one of the most important readouts. Kiesgen and colleagues reviewed this in 2021, and these are 4 common examples of assays that can be used to study cytotoxicity, such as chromium release assays, bioluminescence assays where bioluminescent cells are killed and we lose this signal, an impedance-based assay where we basically look at the confluency, but also again FACS analysis, in this case on the target cells, to see apoptotic markers such as annexin 5 and 7-AAD. So as I said, and as was also introduced in the first webinar by Gera, we use an image-based approach. And for this, we can use the PDO and PDXO material that has already been wonderfully described by Marrit, and see this in 3D extracellular matrix. And what we can subsequently do is add immune cells and any compounds of interest on top of this 3D hydrogel. And over time, this will lead to the migration and subsequent killing of the organoids, which we can assess by 3D imaging, image analysis, and 3D reconstitution. So what kind of readouts can we get in this assay? Well, first of all, since we are segmenting also all immune cell objects, we can look at T cell proliferation. Does the drug treatment have any effect on the numbers of T cells that we see in each well? Furthermore, the T cells will also migrate. And since we do 3D imaging, we can assess where the T cells are in the well over the course of time, as is also shown here in this video. Furthermore, we can also look at tumor infiltration by overlapping the masks for the immune cells with the masks for the tumor objects, and also tumor killing by measuring the volumes and the morphology of tumor objects. Finally, in our assay, we can also collect supernatants, which allows for cytokine analysis, such as that's shown for interferon gamma here. So here's an example of one of our assays in testing T-cell engagers. We can already see from the segmentation mask, which outline the tumor objects, that with an increasing concentration of this T-cell engager, we see a reduction in the tumoroid content in the well. When we look at an early time point, we can also see that this T-cell engager really recruits the T cells, as we see a dose-dependent increase in infiltration, especially for the second T-cell engager. And then when we look later, we can also appreciate that this recruitment also leads to killing, as we see for both T-cell engagers measured here, that we can observe this killing. What is maybe interesting to mention, and that this is in the allogenic setting, so not yet in the autologous setting described by Marrit. And although we do see some effect of adding allogenic T cells to this culture, we can still nicely measure the effects of such T cells therapeutics in an allogenic setup. Of course, we are not the only ones working on this, and the popularity of 3D culture is becoming more and more common, as can also be seen, for example, in this paper by Dang and colleagues, where they used a novel bispecific antibody, which was specifically designed to induce killing, but not to induce any interferon gamma secretion. And we can see nicely that also in their analysis, the organoids are being killed, as also measured by cytotoxicity, by the number of lysed cells. But we can see also that they combine this with the cytokine readout, where their new novel compound doesn't show any interferon gamma increase, thereby preventing unwanted effects of the bispecific antibody. Furthermore, we can also test in our setting engineered T cells. This is an example of an engineered T cell that only acts once an activator is given. And we can nicely see that just adding the engineered T cells, we retain most of the organoids, but giving the activator, we can almost see a complete killing, which can also quantify in the total organoid volume measured here. And one huge advantage of such systems is to have the ability to test many combinations. And that was also shown nicely by Patterson and colleagues this year, still published recently. They use not a 3D culture system, but a microfluidics device where cancer cells are cultured in suspension culture. But by having such control and low amount of cells needed for each condition, they were able to measure the combination of carboplatin, CAR T cells, and also checkpoint inhibitor, anti-PD-L1. And this really shows the strength of preclinical testing with imaging and high throughput systems. Finally, many therapies are directed to be checkpoint inhibitors. We have also tested this in our allogenic co-culture system. And while we see that interferon gamma production is increased upon treatment with Keytruda, a checkpoint inhibitor, compared to its control, we were not able to see any effects on the tumor killing. And this can have multiple reasons. But I first will show you an example of an in vitro technique where they were able, and explain there what maybe we can develop later. So one fact that is well known is that not every tumor will respond also to checkpoint inhibitor treatment. A well-known reactive model is MC38. And what they have done here is to take syngeneic tumors, meaning that they have both an immune and a cancer component, much like the autologous system that we are trying to set up. And by, again, doing microfluidics, they could do live death imaging and see that indeed in this model, anti-PD-1 treatment really reduced tumor outgrowth. So Marrit already described that for now the autologous system also is not yet recapitulating the immune checkpoint inhibitors, although we might just not have found the right model yet. But another thing might be that the T cells are not in the right activation state or exhaustion state. And Pirouz will talk more about that. Moreover, maybe the tumor microenvironment is not correct for us to observe these checkpoint inhibitor effects. However, we have a primary tissue platform, which will be discussed in webinar 3, where we really see those effects that we also observe in the clinic, where roughly 20% of patients or samples show reactivity, also based on the cancer subtype. So that will be discussed more in webinar 3. And so for now, I want to thank you very much for your attention, and I will hand it over to Pirouz. Thank you.
Thank you, Saskia, for introducing me. Hi, everybody, and welcome to in vitro recapitulation of T-cell exhaustion, T-cell dysfunction assay. So the T-cell exhaustion or T-cell dysfunction negatively impacts different aspects of stages of this cancer immunity cycle. And it does impact the T-cell priming, tumor killing, and influences negatively the migration and infiltration of T cells into the tumor, and does it basically by contributing largely to the suppressive tumor microenvironment. To better explain the T cell exhaustion importance and impact on drug R&D in preclinical studies, I want to remind us that the T cells, antigen-specific T cells or tumor-specific T cells, are generated in lymphatic compartments, for example, draining lymph node, and they're expanded, and they are expanded based on antigen-presenting cell information that is coming from the tumor deposit, but they haven't really met the tumor yet. So assays that we do, we have to pay attention that there are T cells inside the tumor deposit functioning and everywhere else. So there's totally a different scenario, very hostile microenvironment in tumor deposit, and that if you don't recapitulate that in your assays, in your assay development, the testing of drug is not complete because it's not showing the physiologically relevant situation. In the tumor deposit, T cells are multiply stimulated by tumor antigens, and this persistence of antigen is something that is different from other scenarios such as acute infection or vaccination. Here, the antigen is not going away and keeps in stimulating TCR, resulting in feedback, a positive feedback mechanism that enhances co-inhibitory molecules. These co-inhibitory molecules then results in signaling that FOXO1 that results in counterpart, which is in this case PDL1, for example, or other co-inhibitory molecules ligands. And then you will have the engagement of the co-inhibitory molecule with the ligand, resulting in signaling that at the end will cause the inhibition of TCR-mediated responses, which we call it T-cell dysfunction or T-cell exhaustion. CAR T-cells also are affected by T-cell exhaustion, both in vitro when CAR T-cells are expanded in vitro and making those batches to infuse to patient. It's a barrier because when CAR T-cells are expanded, they have resulted, or they may result in expanded CAR T-cells that are exhausted or dysfunction. And also same thing, same barrier or challenge in vivo when these CAR T cells are infused into patient because of same mechanism of multiple stimulation of T cells with the same antigen over and over can result in T cell exhaustion. So it's important to address this and to develop assays that resembles or recapitulates the tumor microenvironment with terms of T cell exhaustion to better identify active drugs that are able to effectively eradicate the tumor. To do that, what do we do at Crown? We have focused on the fact that the trigger or initiator of the T cell exhaustion or dysfunction is multiple stimulation. Here I'm showing you 4 rounds of stimulation with anti-CD3, anti-CD28. CD28 is to prevent anergy and focus on T cell exhaustion dysfunction generation, and the scenario should be focusing on multiple stimulation with signal 1 and signal 2. The success of this process to induce T cell exhaustion or exhausted T cells or dysfunction T cells relies in 2 sets of readouts. One is those readouts of co-inhibitory molecules that are associated with T cell exhaustion, enhancement of those. So elevation or augmentation of expression of PD1, TIM3, LAG3, CD30, and these novel ones, which is CD38 and CD39 in particular, is associated with terminal exhaustion. So that is one readout, sets of readout. The other readout is the key cytokine for functionality, to see if the functionality of the exhausted T cell is compromised. So for that, we focus on key cytokine expression and also killing as one of the final verdict of compromise or impaired functionality of these T cells. So 2 sets of readouts. We conduct studies to analyze our stimulated T cells multiply. So as you see here, the first sets of study result that I'm showing is showing the PD1 and TIM3 expression in total T cells, and it's clear that they are double positive for both co-inhibitory molecules, especially after 2 and 4 rounds of stimulation, in vitro stimulation. Then we move on to show the subset CD4 and CD8 and the expression of these co-inhibitory molecules, including PD1, LAG3, TIM3 in these subsets, and showing that the multiple stimulation results in overexpression of those. The next step, we go to show the CD38, CD39, which are the more novel biomarkers of T cell exhaustion and associated with terminal exhaustion. And the trend of kinetic that we see is exactly what has been reported in the literature, which CD39 comes later. Next, we go for functionality. I'm showing the result of interferon gamma, which is the last cytokine to be inhibited in T cell exhaustion, and TNF alpha, which is more earlier. For functional characterization of cytotoxic killing, we use two different methods. One is an allo co-culture system where we stimulate repeatedly the T cells like we did, and then co-culture with the allogenic cancer cell line human, or we use a different system, which is a high content imaging readout based tumoroid 3D culture system where we have the tumoroid and we also have the T cells stimulated the same way, co-cultured. And in two different systems, the result is consistent, showing that four rounds of stimulation of T cells results in compromised killing and reduces the cytotoxic killing. So the summary is that we have a validated, optimized, and validated assay using human healthy donors, PBMC T cells purified from them, and that shows the key features of T cell exhaustion, T cell dysfunction, which are co-inhibitory molecule elevation of expression of co-inhibitory molecules, and also impaired functionality. But not only we show that with increased expression of this cytokine, we show the same kinetic that has been reported in the literature. In that, for example, CD39 comes later after 4 rounds of stimulation, and same thing for interferon gamma, which comes later after four rounds of stimulation. We also show the killing in two different assays, and I think this assay we're excited and ready to test IO drug candidate to see if they have been shown to work in assays that are tailored for not necessarily inside the tumor deposit. Are they also able to be active and function in this assay? Next, I'm going to talk about an assay called T cell modifiers assay. This assay is basically a conditioned and improved version of recall antigen assay. In this assay, we have two settings. One is for CD4 T cell modifiers, and one is for CD8 T cell modifiers. The assay is that we have published data on this assay using small molecules for adenosine as a positive control to inhibit antigen-specific T cell response, and also we have shown the effect of biologics to enhance the antigen-specific T cell response. And we have been able to optimize it for CD4 and CD8. So if you have drug candidates that you want to know if they're more effective on CD4, we have an MHC class 2 recall antigen validated and optimized using tetanus, and we have CD8 using different recall antigens such as flu or CMV, or we have also used HBV in this setting. There are different recall antigens can be used, but I'm showing the result with CMV and tetanus. For co-inhibitory part of or other side of immunomodulation, we have used adenosine as a positive control. If you look at the third column, you see that the recall antigen assay responses is dramatically reduced and inhibited, whereas other approved drug or derivatives of the drug had no effect. So this assay also is T cell modifiers assay is a human-based recall antigen assay that can identify active immunomodulatory regardless of if they're a co-inhibitory or co-stimulatory drug candidate. We have validated this assay functionally using small molecule and biologics. So today we discuss the patient-derived tumor organoid and the power of high content imaging to assist you for testing your drugs in this cycle with terms of T-cell priming, tumor killing, migration, and infiltration. And we also have shown an assay that focuses on the suppressive tumor microenvironment by recapitulating and modeling the T-cell exhaustion to better assess or characterize drug candidates for their anti-tumor activity. Thank you very much. Looking forward to see you in future discussions and webinar.
All right, fantastic. Well, thanks so much, Pirouz, Marrit, and Saskia for the great presentations. We'll now move into a Q&A session. So as a reminder, if you do have a question that you'd like to ask, you could submit it in the Q&A panel on the right-hand side of your screen. All right, well, let's jump right in with a great question. And Marrit, this one will be for you. So what type of information does Crown Bio provide on the organoid models available in your organoid collection?
That's indeed a great question, and I didn't touch on that very deeply. So we do a very extensive characterization of the organoids, of all of the organoids that we have in our biobank. Most importantly, we do a full NGS characterization. So we do a whole exome sequencing and RNA sequencing, and all of that data is actually available to everyone on our Crown Bio website. So feel free to go to our organoid base, subscribe and browse. Next to that, for not for all, but for many of our organoids, we also have some standard of care data. We have some histology data, some pathology, some ploidy data. So there's a lot of data to browse through to be able to select for your ideal model.
Great answer. All right. I think, Saskia, this next one can be for you. What are the advantages of imaging based on readouts, such as the high content imaging platform, compared to fax-based analysis of T cell therapeutics?
Yeah, thanks for that great question. So I think one of the main things that we have also seen in the presentation is that we cannot only address one of the processes that happens in the cancer immunity cycle, but actually multiple of those. So we can directly measure proliferation of the immune cells, but also their migration and their infiltration. And although some of those are possible by fax, that becomes much more complicated in general. And so to really keep everything as is in a 3D matrix in the well really gives a very physiologically relevant readouts, we believe.
All right, fantastic.
And if I may actually add to that, indeed, so that's on the T cell biology, let's say, and as well, if you do the co-cultures and you're working with organoids, then you're also allowed to actually maintain the organoids in their environment. So you don't have to break them out of the matrigel and break them into single cells, which some of the organoids are very happy to, and you're able to actually see their behavior. But some organoids cells will just die in the process of making it into single cells, which will then make it harder to analyze the dying due to T cells.
Exactly, and indeed also to preserve the morphological features, which we also have many different morphologies, right, between different models that can also really be of added value there.
Yeah.
Excellent. Yeah, thanks a lot, Marrit and Saskia. All right, Pirouz, here's one for you. Is T cell exhaustion a different process from T cell dysfunction?
Well, it is a bit. There are overlaps. The T cell exhaustion is a set of series of different stage of suppression through positive feedback mechanism, T cell dysfunction is a little bit broader spectrum. And for example, in our assay, we don't have epigenetic data yet. That's why and people in literature, they tend to use T cell exhaustion slash dysfunction because really shaping the definition of different stage of suppressed T cell or exhausted T cell is not, there's not consensus yet, and for different stages. So to be fair and to be accurate, we can't say 100% the the T cells that are suppressed in our model, or people cannot say it. So they use T cell dysfunction in these cases. When you have all element or parameters accurately defined, it's not, that is the time to better focus and on naming your exhausted T cell, suppressed T cell exhausted, for example. So in general, there is overlap in these two terms, but they both of them are separate from energy, unresponsiveness, senescence, and tolerance. So with that term, they're very close.
Fantastic, great answer. All right, Marrit, one for you now. So how sure are you that the organoids reflect the patient tissue, especially for the PDXO models? Are there any QC steps to check this?
Definitely, yeah, thanks. So there's many QC steps in our processes. So of course, the PDX biobank is extensively QC'd and checked towards the original tumor, and PDX models in general have been, as a golden standard, of course, been looked at very carefully. So the histology there overlaps very well with the patient histology, and then of course what we do after generating the organoids, we also compare the histology of our organoids towards the PDX models as much as we can. On top, as I just already, it's nicely actually following up on the previous question, we do all of this NGS analysis where we also compare, of course, towards the PDX models so that we know for sure that the population of cells that we're growing is indeed the same tumor as the ones that we're growing in vivo. And we have a very nice in-house generated NGS-based SNP paneling that we do to check first of course, for that we're growing the right patient material, but also to check for mouse content, which may actually grow also in PDX-O models. So that's a very renowned problem in using models from PDX models. And we can prevent this by checking our samples regularly on mouse content. And we can actually detect up to 1% of the mouse content, so we can very nicely keep this as low as needed for our assays. And a similar thing, in fact, for the PDOs, where all of these tumors, whenever they are brought in, they're also a piece of the tumor is, of course, preserved for immunohistochemistry, and they're aligned with the PDOs and also SNP'd compared to the original tumor. So yes, extensive QCing.
Yeah, fantastic. Really nice to hear. All right, we'll go to one for you, Saskia. So what is the scale or the high throughputness of these reconstituted in vitro assays?
Yeah, thanks for that question. I think that's really one of the advantages we can offer. We are working in the 384 well plate format. And so basically, and as Marrit has also explained, right, the organoid expansion is highly suitable to generate very large batches as well. So basically, we are limited by, for example, the amount of immune cells we get from a single Buffy code, but we can run many plates with hundreds of conditions in principle in our screening. So it's really, yeah, we also run a lot of very small studies, but also very large studies where we also, for example, look at 10 or 20 different organoids models, even with the same batch of PBMCs. And due to our use also of automation, we can also run that in a very consistent manner, and also we can make sure to use the same batches from the starting material also in multiple phases of the project. So that makes it very scalable to first, for example, set up the conditions we need and then run a very big screen with hundreds of compounds.
Yeah, that's a really great point. Thanks, Saskia. I think in the interest of time, maybe we'll just have one last question. And Pirouz, it'll be for you. So can the T-cell exhaustion assay be used for CAR T-cell therapy?
Yes, CAR T cell undergo the same process of signaling and stimulation as, in a way when it relates to T cell exhaustion as other T cells. In that, they also initiate this feedback mechanism after multiple stimulation, and what we can do here is to simulate by translate this assay exactly to the same way that we do it for with the beads. We can use it with cognate antigen of the CAR T cell, and we can just recapitulate the same scenario that happens in vitro in T cell exhaustion for CAR T cell. For expansion of T cells, it's a barrier, and also for in vivo T cell exhaustion that happens in host. It can be recapitulated in vitro and testing different drugs to see if you can delay this exhaustion process, or you can reverse this exhaustion process. Or it can be used maybe as a batch release. Sometimes that's a determinant of efficacy of a batch of CAR T cell. Eventually, that's doable.
Great, fantastic. Well, Marrit, Pirouz, and Saskia, thanks so much for sharing all your insights today. It's really been a pleasure having you with us today.
Thank you very much.
Thank you.
Our pleasure.
Yeah, awesome. Big thanks also, of course, to the audience for joining us today. So in closing, we hope you enjoyed this Crown Bioscience, scientist.com, and Inside Scientific webinar, and we hope to see you again next time. Have a great day, everyone.